Well, thank you. While we're just getting set up, thanks for the invitation and the opportunity to, this is my first time to New Zealand, so it was great to have this opportunity. So in Canada, we're just starting to go through a process of coming to terms with our colonial history. And one of the ways we do that is with a land acknowledgement. And so I just want to start by acknowledging that the university that I work on sits on the traditional lands of the Anishinaabe and the Haudenosaunee. And I think we have a lot to learn about the history of the people of Canada, what we now call Canada. And I think, it's also at the same pivotal time in our cultural history where we're really starting to think about these questions of inclusion and the sort of long arc, not the positive arc of justice, but the long arc of colonial history, that the influence that colonial history has had on the way we think and do things, even as far as the way we think about science and how we work as scientists. And for me, this has really been helpful in developing a more inclusive and equitable lab environment. And so I just want to acknowledge all the wonderful students, postdocs who I've worked with just in the last 6 years or so at Queen's University. And in particular, I want to acknowledge the students who have contributed to the work that I'll talk about on the left here, as well as many of the collaborators who have influenced this work. And I think, it's not just about thinking about the cultural issues, but also really deeply reconnecting with what it means to be a scientist and the value of traditional knowledge, which is really something that is emphasized in the indigenous communities. And I want to point out at the bottom here, there's this Anishinaabe oral tradition of the 7 grandfathers, and 2 of them in particular, the humility. I'm afraid I don't know how to pronounce these, so I don't want to butcher them, but the teachings of humility and respect, I think they're really the things we should be thinking about in the current context of global change and the impact that we have on the environment. Certainly this history of colonialism is part of the reason why we're facing these challenges now. But I also want to start with a simple observation, which is that for the last 30 years, we've seen that populations can evolve rapidly in these challenging environments, and yet at the same time, species have distributions. No species is distributed everywhere, and throughout the range of a species, those population levels may be variable, dropping to 0 at where we define range limits. And if you think about this from an evolutionary perspective, if all these populations can evolve to local conditions, why do species have range limits in the first place? Why don't those marginal populations just adapt to those marginal conditions, and so that species keeps spreading? And I think, in the context of global change biology, we can look to extinctions and invasions, these sort of dynamic distributions, as opportunities to investigate what these genetic factors are that determine species distributions. So this is just a paper we did a few years ago looking at, just reviewing a lot of the genetic mechanisms that are common between invasions and extinctions, even though we often think of them as opposite phenomena. And we use this metaphor of the looking glass, where the same set of genetic factors are mirrored. But a lot of this is theoretical. We don't really have a lot of deep understanding of how important these different factors are in natural populations. And that's why I've been interested in studying invasive species in particular. And you're all familiar with images like this, where we have species that were introduced to a new environment, and despite having no evolutionary history in that environment, they're able to establish and propagate, and often are so successful that they start excluding natives. And again, the question is, what's the genetic basis for these explosions? So this is the idea behind the field of invasion genetics, which really goes back to this book in 1965, edited by Herbert Baker and G.L. Stebbins. And you can see some some of the famous names here you might be familiar with in terms of different aspects of evolutionary theory, ecological theory, and these are all people who contributed to the book. And so 50 years later, we held a symposium, and there's a few people in this room who were there, where we revisited this idea of the genetics of colonizing species in sort of the modern parlance of invasion genetics. And the basic idea is really just using the tools of genetics, not just genomics, but also the sort of classical statistical genetics that we can do, crossing breeding designs and those other kind of genetic experiments to try to understand this phenomenon. So I'm going to focus on this flow chart in terms of, I forgot to set a timer so I don't go over, just in terms of thinking about the genetic factors that may play a role in determining how species become invasive. And we can start at the top there where we might have a set of traits or genetically based traits that preadapt particular species to become invasive. So if you have the right set of traits, you're more likely to become invasive. And then at the next level, there might be some sort of ecological advantage through the loss of natural enemies or through these maybe some allelopathic chemicals that are produced. And then finally, the third step would be the ability to actually adapt to local conditions within the introduced range as these species spread over these into different environments. And if all of these things are true, we might expect those plants would be the most invasive and maybe to a lesser extent if they don't all hold true. So this first idea of preadaptation probably owes, at least in my thinking, to this paper in this original book by Herbert Baker. His contribution to this book was about what makes a good weed, so thinking specifically about plants, but basically summarize these set of traits. And if you look at things like rapid growth rates, seed longevity, ability to reproduce without specialist pollinators or when populations are small, those kinds of things. So it's a very intuitive kind of logical argument. And this sort of sparked a whole range of studies looking at sort of predictive models, using statistical models to predict which species might become invasive. And you could imagine the management value of that if we could just look at the characteristics of a set of species and figure out which ones would become invasive. We could use that to define what to let into the country or not. And then at the next level is this idea that when species are introduced, they gain some sort of ecological advantage. And this is the example I've given for purple loosestrife, which is one of the species, a plant species that we do a lot of research on. It's native to Europe here, and then it was introduced to North America, and it's been able to do quite well in North America, despite having a much shorter history on this continent. And so some of the ideas here are that when this plant was introduced, it left behind this specialist beetle, the Scaloricella genus of beetle, which includes a few species that feed on purple loosestrife and some other biocontrol beetles that are specialists, as well as potential pathogens. And by leaving those behind in the native range, that allows the species to presents an ecological release that might account for the spread of the plant. And then this is the evolution of increased competitive ability, or EICA hypothesis, which is built on top of the enemy release hypothesis. And it's the idea that once you lose those natural enemies, you no longer have to invest into costly defenses, and so selection might favor plants that no longer produce those defenses, but instead invest more into growth and reproduction. But then there's this other part, which is as this plant was introduced to the East Coast and then spread across North America, there's quite a wide range of environments that are represented here by this one species. With the main one that we've been focusing on is differences in season length, which there's almost a 2-month difference in growing season between the northern populations and the southern populations. So this leads to two main questions that I want to address today. Thinking about this question of invasion genetics, and because we're in starting the invasion vectors and pathways, I want to bring that in. But the main questions are related to those three stages. What traits make species invasive? How common is adaptive evolution during invasion? And we can think of that both at the intercontinental level when they leave behind natural enemies, but also as they spread across North America, those sort of very localized level of adaptation. And we're talking about vectors in this session, so we've got to think about the role that human activity has played in shaping these patterns that we might be using for biological inference. And so this is we'll start with this idea that human introductions represents a sort of null hypothesis for thinking about some of these questions. And this is just an image from Wikipedia showing the connectivity of global shipping traffic. So which traits make species invasive? So when I was a graduate student, I read these papers that were actually based on bird introductions to New Zealand, and they showed very clearly that there was a very good predictor of which birds would become successfully established in New Zealand, and it was basically the birds that were introduced more, the ones that people put more effort into introducing. And that got me thinking, if there could be an effect where if people are choosing what's being introduced, then the traits that are associated with those species might be the traits that we detect. And it's not that there's an ecological, it's not that those traits make the species successful invaders, it's just that those species tend to have traits that either are valuable to humans, and that's why they're introduced, or just humans are non-randomly selecting species, and so the traits that they have will be different. And so my colleague Ian, who's actually at this university, Ian Duggan, somehow came across this obscure monograph from 1943 where they had basically reviewed all the salmonid introductions to the state of Nevada. So we had this very sort of historical this historical account of all the different introductions. So which species were introduced, where, how many, eggs were included. And so just looking at, just sort of codifying that written history as data, it was pretty clear that the species that were successfully established in Nevada, in this state in the U.S., in the black bar here, they had more attempts at their introduction, and then even the average number of propagules per introduction was higher. So again, it's like we saw with the New Zealand birds, that those salmonids which established were the ones that were more effort was put into establishing them. And then we looked at, or I looked at what the different genera were here. So these are 5 different genera. And then on the right, this is the total number of species at the time. This was from a database, a fish database. And how many of those potential species were actually chosen to be introduced? And you can see that there's a non-random selection here. It's not, for example, here we've got 14 species of Oncorhynchus. About half of them were introduced, whereas, so that includes things like rainbow trout. And then in the case of Salmo, there's 27 species, but only 2 were introduced, so a much smaller proportion. And because of this non-random sampling of different genera, we could compare the traits of the established and failed species as well as just the global species pool. And there's a bunch of traits here like size, weight, age, latitudinal, temperature range, things that you might make biological hypotheses about why these species are successful, but it really just comes down to the ones that were introduced more are a non-random sample. And so the traits that differ are most likely just due to the fact that these were the species that were chosen to be introduced in the first place. So that's the idea of the propagule pressure as a null hypothesis, and then we kind of formalize this in a paper in Biological Invasions where, you know, we think about all the different steps of becoming an invader, from introduction to fully invasive, widespread, abundant populations. And you know, these different stages may involve different kinds of filters, and so we end up with a different set of species that's not really due to the sort of ecological hypotheses that we're used to thinking about with the traits of invasions, but they're more about the human element, the human factor. And so that was kind of the point of the paper, is why this is important to consider. So we're going to sort of continue on that theme, but now we're actually going to talk about evolution, except that we're going to think about the different levels of evolution. So again, either intercontinental, so the introduced versus the native range, or among introduced populations within the introduced range. And so we did this meta-analysis a few years ago where we just took these the common garden studies of plants. The nice thing about the plants is, you know, they stay where you put them and you can mix them up however you want. So lots of, well, at the time there were 33 different studies where people would take plants from the native range and the introduced range, grow them in the same environment to look for genetic differences. And those studies were more interested in this native versus introduced comparison. But in this case, we just asked, how variable are these populations within each region? So if we measure the variance among population means, that gives us some idea of how variable the phenotypes are among populations in these common garden environments. So we're really talking about variation for quantitative traits here. And on the x-axis, this is the variation among native populations. So this is all the population divergence that has occurred. You know, if you think about all the things as these plants have spread northward. In many cases, we're talking about Europe and North America. So it's post-glacial expansion, thousands of years of evolution, and this is the level of divergence among populations. And then you compare that to populations in the introduced range, where we're talking about a few decades, maybe a century or two max. And you can see that the level of divergence among populations for these quantitative traits is very similar between the two ranges. It's almost, in many cases, it's higher in the introduced range than in the native range. And then, excuse me, not only that, but these population divergence is not random. It often falls along environmental gradients. And sometimes this can make it very hard to figure out whether this is an intercontinental change or just a local adaptation or even drift and bottlenecks, but happening at the very local level. And there's 4 examples here where it's, so we've just coded I think the native in white and the introduced in black. And if we just try to fit, if we just use in this meta-analysis just fit latitude or latitude by region interactions, we get significant Clines in 14 of the 19 species. And what's important here is that most of these studies were not designed to detect Clines. These were just designed to compare native and introduced populations. So the fact that we detect this significant effect, even though region is a factor in the statistical models, it suggests that these Clines may have very important consequences for thinking about the evolution of introduced species. And it's tempting to think that these might be adaptive because we're talking about latitudinal gradients, where we're changing photoperiod, temperature, and whatever, biotic stress. Lots of things can vary with latitude. But we can also get Clines through non-adaptive processes. And this is a very simple example of a quantitative trait model where we're just looking at stepwise colonization along a gradient. And in this model, the alleles are not under selection. So any Clines that we find in this model are not adaptive Clines. And what we do is we imagine subsampling 20 populations along this gradient. And then we've got the different scenarios here. So between 5 to 500 founders in each event, and between 5 and 500 loci. And it actually doesn't matter how many loci, or how many founders we have. The number of significant Clines, if we just fit a linear regression to those Clines, it's always about 33% that are non-significant and 66% that are significant. And in fact, half of them, 50%, are significant, highly significant at the p less than 0.001. So just this subsampling, this is basically genetic drift playing out in space. And so we expect that Clines may be quite common. Non-adaptive Clines can be quite common. And then the question is, what's the implication? So what's the implication of these Clines for whether they're adaptive or not, for comparing phenotypes or genetic differences for phenotypic traits between the native and introduced range? So we did another simulation here where we imagine local adaptation or local Clines, either adaptive or non-adaptive Clines in both the native and introduced range. And then we went back to these original common garden studies and used those latitude and longitudes in this simulated data set. And we just asked how often would we find a significant difference between native and introduced populations in this simulation where we know there's no difference between between regions. So there's no difference between the native and introduced range, but within each range we have latitudinal Clines. So using these actual sampling locations, we found significant Clines 80% of the time. We found significant differences between the native and introduced range 80% of the time, even though it didn't exist. This is if we ignore the Clines. And we just ask what's the mean difference between the native and introduced populations. So 80% of the time we get a significant difference. Half the time we get a highly significant difference. Even though that difference is not there, it's just because we haven't included latitude as a covariate in the analysis. And the reason this is happening is shown here. So this gray represents the 95 and 99% confidence interval for a given number of populations per region and what the expected difference between the average of the native and introduced locations are. And you can see that the significant in red, highly significant in orange, a lot of these are actually falling inside the null expectation. And that's because when we go and sample populations, we often don't have a purely random sampling design. It's often opportunistic. And if we don't account for the spatial covariance in that data, then we end up with this high rate of false positives. So that's another way that we need to think about a neutral model when we want to compare native and introduced populations. That evolution that we attribute to the large-scale introduction to a new continent might be just an artifact of local scale evolution that we're not accounting for in our analysis. And here's another example. If we think about doing a QST-FST analysis where QST is our measurement of trait divergence, sort of like that first figure I showed you, and then we have FST as our measure of molecular divergence. Even under this scenario where the populations are in mutation selection equilibrium, we'll go through a few scenarios here, but let's imagine you have a single source in the native range, or maybe a few sources, but they're all from similar environments. That genotypes are introduced into different environments along a gradient, so these different colors represent that environmental gradient. So we're going from a single source to multiple destinations with different environments, and then adaptation is occurring across those environments. So depending on whether we're comparing each population to its origin, or we're comparing the introduced populations against each other, we can often end up with counterintuitive results here. So the second scenario, for example, if we have populations that have evolved differences in the native range, and then we have multiple introductions into more similar environments, then we get these different predicted QST, FST results. And then here's the last example of parallel introduction. And so it's not always clear, just with a purely this kind of observational study where we measure quantitative trait variation and molecular variation, for the same phenomenon, we can get different results depending on which populations we're comparing. That's really the main take-home point here. So then the question is, how do we even know if adaptive evolution is important in invasion? And that's where we've done some experimental work with this species I mentioned called purple loosestrife. And it's a really nice system. I was telling someone, I think it was Neil earlier, I actually started in aquatic invasions as a master's student, and I did a PhD in plants, and it turned out to be such a nice experimental system that I still use it now. And one of the nice things is the way the plant grows, every year it dies back in the winter, and then either regrows from roots or seeds. And so at any given point in time, we can look at a plant and know its phenology, because we have the vegetative growth from the base, from the ground to where the inflorescence starts, and then we can easily identify the different parts of the inflorescence representing buds, flowers, and fruits. And as the plant is developing, it first develops buds. Those buds then open to make flowers, and then those flowers get pollinated to produce fruits. So we can use this to understand the phenology. And one of the most striking things in our common garden experiments is when we grow, these two plants are grown in the same environment near Toronto. These are northern plants that are almost done flowering, and this is a vegetative growth of a southern plant that hasn't even started flowering. So we get these big differences, which we can observe in these common garden experiments. We've done a bunch of these. Here's just a quick little video to show you what it's like harvesting one of these experiments. So these are two of our graduates, one former graduate student and one undergraduate, harvesting some of the plants from one of these big common garden experiments. Here, this is one of our blocks, experimental blocks, and as we zoom out, you can get a sense of the scale of this kind of experiment and what it can tell us about, adaptation in natural environments. Okay, so one of the key experiments that we've done is, what we call a reciprocal transplant. And that's where we take genotypes from different locations, like up here in northern Ontario. Here near Toronto, these just focus on the squares. And down here in northern Virginia. And then we grow all the genotypes at 3 locations in the north, mid-latitude, and south. So at each location, we have the local genotypes that are matched color. And then we have the foreign genotypes that come from a different location. And so we published this a few years ago, showing that these populations are locally adapted. But what's really interesting is the way that local adaptation has evolved here. And so this just shows, these columns are 3 different environments, in the northern environment, mid-latitude, and southern environment. This is our model of what selection looks like in those 3 environments. And then this is our observed fitness data. So in the north, in this environment, the north would be the local genotype. If we move that to more southern gardens, you can see that the fitness stays the same. In the south, this would be our local genotype. As we move it north, its fitness goes towards 0. And what's happening here is that those plants are delaying reproduction, which is good when you're in a long growing season. But as you move to shorter growing season, you don't have time to mature offspring. And so the way we get local adaptation is only one of the genotypes has this changing reaction norm with environment. And that was a lot of work. You saw that one experiment at our field station, and this was at 3 sites. But it's still only a small part of the range in North America. So we got this idea to use what we knew from these common garden experiments to recreate evolution from herbarium records. And you can see here, so Kentucky, if you're not familiar, it's somewhere, I don't know, is that right? Somewhere around here. And Wisconsin is up here. So we're talking about a more southern and a more northern. So these were collected on the exact same day of the exact same year. This one in the south, this is almost all fruits, so it's almost done flowering. This one is just getting started up in Wisconsin. And so we use our what we know about the development of purple loosestrife. So each purple point here is a herbarium record, and then each of those gray points is a local climate station or weather station so that we could recreate the growing season in that year of collection. So, for example, if we just divide this into East Coast, which is the source of invasion, then spread to the Midwest, then to the West Coast, you can see that the Clines that we get are strongest in the East, where it's been been established the longest, and they're weakest in the West. And not only that, but we can go back through time. Sorry, I'll go through this quickly, but just looking at how Clines evolve, we tested whether it was a relatively constant rate of evolution, going from black to red, or sort of this punctuated model where rapid evolution occurs really quickly and then slows down. And this graph just shows that it's the latter. These are just parameters for our model, but you can see that the rate of change is pretty constant for about 100 years and then levels off for these parameters. So consistent with that second example. And then I just want to mention that we are doing some genomic work, so I know I've been kind of critical or cautionary about using these neutral models and null models, but I certainly think genomics have an important role to play. And here's just an example where we did a differential expression analysis of early versus late flowering, and some of the really highly expressed, highly significant genes, there's no matching similarity in GenBank that we can find, and so we're really interested to figure out what those do. And then I'm going to skip over this, but just some modeling related to the genomics of local adaptation. But I'll just conclude by summarizing that if we want to understand what traits make species invasive, we have to think about propagule pressure, the role that human activity has played. And when we want to compare between the native and introduced range, we have to think about local processes. And I'll just leave it there. There's just a few more genomic things. But hopefully that's a good example of how invasive species are not only important for conservation issues, but also can tell us something fundamental about ecology and evolution. Thank you.
So when it comes to making or kind of trying to come up with a null model that you want to test for adaptation with, for instance, if you're trying to actually identify a Cline, do you think that if you have, say, some subsetted null SNPs or something like that, do you think that if you find a Cline because what you were talking about was that a Cline might explain some sort of adaptive difference, but if you actually are looking for a Cline in itself, do you think that it would be likely that you could have caught that if it was null in a null model, or do you think that's a slightly different scenario?
If I'm understanding, I don't want to go back to this
slightly badly phrased.
I just want to make sure I understand, but you're thinking about Clines at the molecular level, so like a SNP Cline. And so that's not unlike what we've done here, in that we're looking at phenotypic change, but the reason we're getting that phenotypic change is we're getting Clines at those individual loci. And so, but you're right that the cumulative effect on phenotype is stronger than the Cline you would observe in an individual locus. And so I think that's an interesting question to think about, and maybe even do some modeling about the trait itself versus a typical locus. Yeah.
I have a question.
All right.
And since there's no other, I will indulge myself. So a lot of invasions are studied post hoc, and you're showing that there are issues with, that kind of work. what would your recommended improvements be if you had the choice to, go ahead and do a large study? What would you recommend be the better way of doing it?
Oh, well, yeah, I don't know if I can generalize, but I would say, I think just showing that you've thought about the potential influence of human activity, or if you're doing these native and introduced comparisons, that you're also considering the local level differences. So I think certainly for those like large scale contrasts we're comparing across continents or large geographical areas, I think we're not really at the point where we're thinking carefully about the local scale process within those regions and the sampling design. And so, we could probably develop a list, but I don't have like a very simple general answer for that. Yeah.